Okay, very good morning to all of you. This will be my last lecture to all of you in this course. But I believe we are going to meet maybe in next year or somewhere in other courses. I just want to remind you again, the topic 1 and topic 2 for you to create MCQ will be by this Sunday. And then you have to create the MCQs for topic 3 and topic 4 by 8 September. And for the question you need to answer and for the comments you need to make, can be any questions, can be at any topics, okay? But you need to make 5 comments and answer at least 5 questions, okay? Answer at least 5 questions, make at least 5 comments. And it's very good. I say the leadership positions changed over time. And this is my last lecture. I want to repeat again, in topic 1 and topic 2, right? We have 2 lecture slots for each topic. And for topic 3 and topic 4, we only have 1 lecture slot. Okay. And this is related to topic 4. Topic 4 basically helps us to understand better about this process. We mentioned all this, right? And I believe today's lecture will help you understand better for what we have already observed, or some of you already asked me about the process in a molecular level or in the detail, okay? For example, how the crossover takes place, how the effector gets integrated into bacterial genome, right? How the transduction happens and the delivered alleles going to replace the recipient alleles, right? How the intragenic mapping or the crossover takes place. We cannot explain all, but today will help you make a little bit, I hope, and you can consolidate your understanding and make connections between the different lectures. We're going to cover 3 different types of the recombination: homologous recombination, site-specific recombination, and the transposition. We'll spend a little bit more time on homologous recombination, which is we learned in the topic 1 between non-cis chromatids, right? There are 2, actually maybe much more than 2 models, or actually mechanisms, or you can say it's a hypothesis to explain how the crossover happens in meiosis, which is at the prophase 1. The first model is called a holiday model. And this holiday, you see there's a double L. It's a longer holiday, okay? Then in this particular model, you observe there are 2 leaks, right? One happens in the 1 DNA molecule. Okay, I wish to make it clear to you because this is somehow confused by actually some of you. You can label this chromatid as 1, 2, and 3. This is 3. Okay, then this is 4, right? If the chromatid 1, 2, 3, 4. And then you look at the string here, and there's a 1, 2, 3, 4, right? It is not the same. Okay, the number 1 and the number 2. The number 1 and the number 2 are cis chromatids. Number 3 and number 4 are all cis chromatids. So the crossover happens between number 2 and number 3 is a non-cis chromatids. Okay, here actually this represents the blue line. The 2 blue lines represent 1 DNA molecule from this chromatid, and these 2 double red lines represent 1 DNA molecule in this chromatase. Okay, so this is actually from chromatase 2, and this is from chromatase 3. Okay, each DNA molecule has 2 strands, right? So don't get confused. These 2 blue lines, actually it's one DNA molecule from one chromatase. Yeah. Then the nick happen in one strand of the DNA molecule. So this nick happened in this one DNA molecule, and this nick happened to this strand of this DNA molecule. So both DNA get nicked, but each just one strand get nicked. Okay. Then once you nick, there's a strand called invasion. Means this is your territory. You go to another strand's territory, right? So this one, so the red go up and blue coming down. Okay, then they form a cross. I need to bring your attention that invasion is initiated by the 3' ends. Means the 3' ends go up, and later one the 3' go down. Okay, so this is a directional molecule. DNA is a directional molecule. Protein also a directional molecule, right? So for DNA you have 3' ends, 5' ends. For protein you have the N terminal, you have the C terminal. They are all called directional molecules. Okay. Then once they invade, they so-called have the branch migration. In this scenario, they move to my right side. Then if, okay, this region, they are not identical, if they are just homologous, they may have different alleles. In this example, right, they have a big E allele, you have a small E allele, you have, then the string gets swapped. So there's this E coming down and this E prime going up. Okay, they create a region called a heteroduplex region. Heteroduplex region means in this particular region, there are some nucleotides doesn't pair to each other. In the DNA, right, A always pair with T, G always pair with C. But if the two alleles, the gene sequence is not identical, when you go to swap, there may be some base cannot pair to each other. So this region will call a heteroduplex. Okay. Once they form the heteroduplex, then they're going to resolve the cross. Means the cross must be resolved. Then there's two different ways to resolve. One is called a vertical cut. Okay, you cut from top down position, showing to the left side. Then you generate the two DNA molecules, which is heteroduplex and with the recombinance. But if the cleavage goes through the horizontal line, shows as an edge, right? Then you generate a DNA molecules which has heteroduplex, but there's low recombinance. What I mean is recombinance. What I mean there's low recombinance. You see here, there's a recombinance, there's a low recombinance. It's because if you look at the original composition, right? So if you look at the original composition here, are all the big letter or capital letter, D, E, F. Then those strains all are small capital letter, but after this cut, then they're going to swap, they change it to the small f going up and the big F coming down. So if you look at the color, right, this one, you had blue and red, this also had red and blue. So that's why they call it recombinant. It's different from the original ones. Okay, if we look at this one, you say at the beginning it's a DEF, you also, then at the later you still get a DEF. It's a blue and a blue and a blue, blue. Even though in the middle, there are some heteroduplex region, this one is considered, consider non-recombinant with heteroduplex region. And because of this one, many students get confused and they very often argue. Okay, this is not because you're understanding just the biological way of the terminology. Okay, so here is called non-recombinant with heteroduplex. And this heteroduplex must be repaired, because there are DNA sequences are not paired to each other. The system, the cell will be able to recognize those kind of the mismatch, then go to repair. And the repair will generate different outcomes. We're going to elaborate later. Even though, for example, even though the outcome become D small ef, D small ef, then this allele get changed, and the big E coming down. So this heteroduplex region, even though they replaced, then it become big D, small e, big F. It's different from the original ones, right? It become recombinant. Yes, it become recombinant. Okay, now it can call it recombinant, but this recombinant is not come directly from the crossover, directly from this kind of the recombination. Okay, but it comes from, it generated from the repairing. Okay, it's after the crossover and generated through the repairing stage. So in that scenario, we still maintain and consider the crossover in this step does not generate recombination directly, but the repairing at a later stage may lead to formation of the recombinants. Okay, I hope. And here is a nice video to show you. Maybe I watch this one. Let me control arrow. Yeah.
The first molecular model of the genetic exchange that occurs between homologous chromosomes during recombination.
I wish all of you watch the video carefully, and you can catch a mistake in the video. Even though this is generated by the textbook, the video.
Was proposed by Robin Holliday. The Holliday model involves several steps. First, the homologous chromosomes are both nicked at identical locations. Then, the strands from one side of the nicks invade the homologous chromosomes, base pairing with complementary strands. The invading strands are next covalently linked to the original strands at the nick site, forming what is called a Holliday junction. The Holliday junction migrates away from the original nick site, a process called branch migration. As it does so, DNA strands are swapped between the chromosomes. This creates heteroduplex regions on both chromosomes, where minor base sequence differences between homologous chromosomes results in a region of DNA with a low percentage of mismatched base pairs. The length of branch migration may vary, but at some point breaks are made in the DNA that end migration and resolve the entangled DNA strands into two separate chromosomes. There are two ways that breaks can be made. One results in non-recombinant chromosomes, while the other results in recombinant chromosomes. If the crossed strands are cleaved by an endonuclease, then after ligation within the chromosome...
So this is the horizontal cleavage I mentioned just now.
The first molecular model of the genetic...
No, not here. Okay, continue here.
A low percentage of mismatched base pairs. The length of branch migration may vary, but at some point breaks are made in the DNA that end migration and resolve the entangled DNA strands into two separate chromosomes. There are two ways that breaks can be made. One results in non-recombinant chromosomes, while the other results in recombinant chromosomes. If the crossed strands are cleaved by an endonuclease, then after ligation within the chromosomes, there will be two non-recombinant chromosomes with short heteroduplex regions. Alternatively, if one rotates one DNA helix 180 degrees, a process called isomerization, we can visualize how uncrossed strands can be broken. After the uncrossed strands are cleaved by an endonuclease, ligation can produce recombinant chromosomes with short heteroduplex regions.
So this is the so-called, the second one is a vertical cleavage, and it generates a recombinance. Okay, we can just maybe look at this one.
Be two non-recombinant chromosomes with short heteroduplex regions. Alternatively, if one rotates one DNA helix 180 degrees, a process called
Okay, any one of you identify an error in this video?
The first molecular model of the genetic exchange that occurs between homologous chromosomes during recombination was proposed by Robin Holliday. The Holliday model involves several steps. First, the homologous chromosomes are both nicked at identical locations. Then, the strands from one side of the nicks invade the homologous chromosomes, base pairing with complementary strands. The invading strands are next covalently linked to the original.
Okay, imagine if this is become a question in the exam, then I have the videos ask you to watch, but of course it wouldn't be. But just imagine, right? Then ask you, can you identify an error in this video? Anyone noticed? Yes, please. Yes, thank you very much. Yes, she identified the error. You see here in the error, as I said, right? I emphasized the invasion is 3 prime ends. It's not 5 prime ends. But in this video, actually, this shows the invasion is the 5 prime ends. So they say if you go back, even though it's a small error, but it looks like maybe the video was created not by a biologist, and maybe they didn't pay really much attention about this.
Invade the homologous
this
chromosomes,
actually the invasion
base
is a 5
pairing with
prime ends.
complementary strands.
Yeah.
The invading strands are next covalently
biology.
linked to the original strands at the nick site, forming what is called a Holliday junction.
Okay, this actually is a mistake. It's not, you see this prime ends is 5 prime ends. And this prime ends also is 5 prime ends. So it's not 5 prime end invasion. It's just a 3 prime end. Should be this end go up and this end go down. Okay. Okay. There's another model we call the double strand break model. And then in this model, you see at the very beginning stage, it is already different from the Holliday model. And this model is considered maybe more accurate, okay, and developed later, means after the Robin Holliday's model, okay. This is called a double-stranded, not double-strand break model, and in short, we call it DSB, okay, DSB. It's a double-strand break model. The 2 nicks happen in one DNA molecule, right? It's not one in each DNA molecule, it's happening in one DNA molecule. Then what happens is, once you create the good resection. Resection means you're going to chop off this cut. Yeah, you cut here. Okay, then here you cut. Then you're going to generate the stick ends here and here, right? So this was mean a resection. Okay, resection. Then by the enzyme, different enzymes in the different cells, then you generate 2 stick ends. Okay, it's 1. Okay, and these 3 prime stick ends going to invade to the lower DNA molecules. We're showing here is a red or purple. Then you see, right? Once they invade, actually they push up this DNA string, so it will looks like a D loop. Okay, push up, and this was pushed up D loop will be used as a template to synthesize this gap. Okay, so this D loop DNA string will be utilized as a template to fill the gap of this region, and this here, this strand will be utilize a template to synthesize a gap between these two. Okay, between these two. Okay, so this is what happened. You see here, you're going to synthesize, right? This will be for this. Okay, data string you must have a, for data string to be synthesized, you must have a template. Okay, then for this, right, this one, it is use this fragment as a template. And by this way, eventually you're going to form two crossover, two crossed structure. Okay, and this two cross structure will be cleaved in many different ways. Okay, here is a little bit complex part of today, and this is one of the important part for today. Yeah. So how the two cross get resolved? They may have different ways. Let me just remove eraser. Yeah, remove all this. Okay. So the first way to resolve is an easy way and easy to observe, which is through this kind of dashed line. Yeah, it means, you see, they cut at the horizontal. The two cross was cleaved horizontally. Okay, then you generate DNA molecules which has this kind of the heteroduplex region, but without swap these alleles, right, in the two ends, right? Yeah, you see, it's still A still go with Z, small a go with small z. So this is a non-recombinance, okay, with short heteroduplex, okay? And this is not a favorite because it doesn't generate any very much genetic diversity, right? Then if this go to cleave in another way, for example, one cross at the cross strand, another one cross at the outer strand, right? And this cross isn't cleaved, and this cross isn't cleaved. So how and what is outcome? Okay, then you will look at the top one DS strand. You move all the way, right? Start from here all the way to this, right? You meet in the first cut. Okay, you move all the way here, you meet the first cut. And once it cut, right? This one is also cut. So this going to cut and swap, as shown here, right? This lower DS strand we're going up, the upper one are going down. So this is how they're going to swap and flip up and down. Then you have this kind of pink region coming to this molecule, and this blue region come to this molecule. And many of you have the difficulty to imagine if this strand was cleaved, and this strand can move up, and this string, but how these two strands get swapped? How these two strands get swapped, right? How this because they didn't cut on these two strands. Okay, later I will show you. But temporarily, let's just look at the second one, right? If this one, you go all the way, and then you meet in the first cut. Let me just remove eraser. Right? If you remove this, okay, all this. If you look at this strand, you come all the way, you look at the, I'm drawing a red line, right? You meet the first cut, and you join to this molecule, this fragment, okay? And since there's low cut, right, you go all the way down to this way. So this is how the second strand, right? How the second strand form, okay? How the second strand form. But some of you observe very somehow attentively and carefully. Then you look at, oh, it should here is it should be the purple color. Why here come a small region of blue? And you see here, yeah, you're all the way down here, all the way down here, and the way down. Should be all the blue. Then why there's here? Because the textbook does not explain very well in this part, how this color changed. It's because they think if here is a purple and here is a blue, if this is blue and this is purple, right? They think they are heteroduplex. They must be repaired to become homologous, become the matched. This match become matched. Okay, so they show the same color. They want to show the same color, just trying to indicate the heteroduplex has been repaired. Okay, similarly here, right? You see the two smaller region of the purple is also trying to show you, okay, the mismatch has been repaired. This has been repaired. Okay, so if you look at downstream, right, it's very much simple. All the way purple, come here, right? Then it's just all the way blue. But here it shows a little bit of purple, right? Right? Here you show a little bit of purple, but supposedly it should be the blue. Then they going to be repaired and show the same color. But of course, here it can both become blue, and here it can both become purple. So long they are the same color, then they are considered as a homolog. Okay, considered as a mismatch repaired. Okay, if there's any region shows different color, then they shows there are mismatch. There are still heteroduplex. Okay. Then, as I mentioned a few seconds ago, right? Then if this cross isn't cleaved, how this cross get resolved? Okay, even though you cut the outer strand, many of you still have to imagine how this get resolved, and that's why I have this kind of video to tell you, right? This is the video actually I prepared. For example, let me show you, Control Arrow. Yeah.
Hi, I'm going to demonstrate Bob's.
When I was very young, okay, when I was very young. Yeah, so this actually prepared this video in my office and I used the paper clips.
Blended brick model. There are 4 strings of paper clips, 2 in pink and 2 in blue. The 2 pink strands represent 2 DNA strands in one chromatase. The 2 blue strands represent 1 DNA molecule in another chromatase. There are 2 non-cis chromatids. I'm going to show you a crossover happen during meiosis. The first, a break is introduced in one. Okay, these 2 is joint.
I skip in the middle part. I just want to show you the 2 cross, how it gets
joint
resolved. Okay.
This joint here. We return it back, there's 2 junctions. Then we're going to resolve it in a different way. One junction is cleaved at the cross strand, but at this point, the crossover strand is not cleaved. Instead, it cleaved at outer strand. And shown here, this is cleaved at outer strand. Means cleave here and cleave here. Then we say how it's going to resolve and how it produces recombinant chromosome. Let me see here, this is their cleave. Then it will cleave here, but it won't cleave here, and one is cleaved here. Pay attention to, at this junction, the cleavage takes place at cross strand, and this one cleavage at out strand. Okay, you see the junction is still there. Then how this junction is resolved? This junction resolved is by rotation of those DNA molecules. And then showing here this blue tape showing you the hydrogen bonding between the 2 strands also same.
Hydrogen
so
bonding.
it's going to rotate let me tell you how you're going to rotate like this. You're going to rotate back, rotate, and this one also going to rotate. So once you rotate, the junction is resolved. Then once they joined here and join here and join here, because these two strands changed, rotate, and this changed back. Therefore, after resolvation, you have big A area for the top DNA molecules, small B area, and a small A and a big B area in the lower DNA strand. So this is how the crossover happen in meiosis may create the recombinants, new recombinants, because you see now there's a small A, big B.
I hope that this kind of demonstration help you to understand better how the crossover get resolved, right? And how the two strands get swapped. Okay, two strands get swapped. Yeah. Okay, so by rotation. Of course, this rotation is facilitated by many enzymes and proteins. Yeah. Okay. For example, this actually shows you the detailed stage. They involve many different types of proteins, shown in the name and the colors. I don't require any of you to memorize the protein names and how they function. Okay. Even though you memorize maybe a few days or even 2 days later, you will forget. Okay, you will forget. So there's no meaning for you to memorize, okay, which protein are involved. For example, these are protein names, these are protein names, all these are protein names are involved. I just want to elaborate at which stage, at each stage, there are many different types of proteins get involved and to facilitate the process. Okay, and this is the one of important part of today's lecture. Okay, one important part of today's lecture. That's why we have the I icon here. I icon here. Okay, and this is another student, right? Once they go back, they watch my demonstration and draw the diagram on their own and confirm with me whether her understanding is correct. Yeah, it's really good. Yeah, it's the way how we learn and internalize the knowledge, right, to your own. So you learn and to watching other people's explanation, then you're trying to explain on your own, right? So if you can reiterate what you have learned in your own words, that means you learn very well. You have a question?
Yeah. If we form a non-crossover product,
Yeah.
do the chromosomes keep going until they form crossover products? Or they're happy?
Sorry, I didn't catch. Repeat again. Yeah.
If you form a non-crossover product,
Non-crossover products.
where you've got the heteroduplex complex,
Yeah.
but they're not recombinant,
Yeah.
do the chromosomes, will they keep going until they get recombinant?
They will keep going, but as I mentioned, right, later, during later stage of the repairing, it doesn't mean they will become recombinant or it will become recombinant. It's a really chance by chance. I will get to mention it later. So that's two outcomes. It may become recombinant. It may keep going. After repairing, it restore back to the wild type.
And if it restores to the wild type, it
Yeah.
stays as the wild type?
It stays as the wild type, yes, yeah. Okay, can you just have a quick try? I don't have the poor EV, I don't care it, just you read the question and you think for a while. I take you some time to digest, right? What we have learned, I think is very important for today. Basically, they try to compare the two models. The early Robin Hornidis model, later the DSB model, double strand break model. DSB model. So A, B, C, D, E, which one is a choice? Let's look at the first one. In Hornaday model, so long as the Hornaday junction is nicked, homologous recombination has occurred. Is correct or not correct? Anyone? If we say it is correct, put our right hands. Nobody. Then assume all of you think is wrong. Actually, the statement wrong is not correct. Okay, good for all of you. Because there are two possible, right? One will generate recombinant, one would then depends on how the junction was cleaved, right? So there's two ways, right? One is cut on the cross strand, which is a horizontal cut, then would then generate any recombinations. Okay. Another cut is called a vertical cut, or is, let's say in the video, alternatively, if the strand gets rotated and the crossover gets cleaved, non-cross strand gets cleaved, then it will generate. So it's not like this. There's two possibilities. And in the question option 2, in the DSB model, recombinant chromosomes are formed when only one Hornaday junction is nicked and swapped to resolve the other junctions. Yes, that is correct, right? Actually, this is how I demonstrated in use of paper clips. And the third one, in DSB model, two strands in the same DNA molecule are nicked. However, in Hornaday model, a break occurs at identical sites in one strand of the both parental chromosomes. That's correct. Okay. And the last one, in DSB model, if both Hornaday junctions are cut, there would be no genetic variation at all. So that is not correct. It's not correct. Because there are variations. The variation is in the heteroduplex region. Okay, there are genetic variations. These variations is in the heteroduplex region. So the number 4 of the option is not correct. Therefore, for this answer, the it will be 2 and 3, which is C is a correct option. Okay. And to connect what we have learned and observed in the topic 1, and here just trying to make you to imagine and to visualize. Okay, this is the at molecular level, and this is what we observed, right, in the process 1 of the meiosis, leptotin, zygotin, picotin, diakinesis, and triplotin and diakinesis, right? So you see, actually, the leak happened quite early in the leptotin, and the crossover all happens actually already in the zygotin, okay, in the zygotin, yeah, and they're going to be maintained until all the down steps, okay, until all the down steps. So if you look at here, right, so in the leptotin, DSB already formed, and at the zygotin, the recombination already take place, and in the picotin, you're going to resolve, okay, yeah, but this issue is when it's going to completely resolve. There's a saying is only until the anaphase 1. Okay, until anaphase 1. Then during the homologous region, right, there are some events may occur. One is so-called gap repairing, another one is mismatch repair, right? So this is the somehow it's events happen during the homologous recombination or after homologous recombination. For example, if this kind of break happen in a region which carry an allele, carry gene allele, here shows a small b, and you have a big B. So in this scenario, if the gap is happened in this particular region, very critical region, deciding this area to be a small b, then after repair, once you use the downstream as a template to repair the gap, then you will change, okay, the smaller b to the bigger b, right? Because this gap, this string was centered by use this one, and this string was centered by this one. So the upper small b allele will be replaced by the big B allele. Okay, so this will become donor, and this will become a recipient, right? The recipient will change into the big B allele. So this is the gap repairing. Okay, this is in the gap repairing. And there's a mismatch repairing also, as I said, right? During the migration, and then generate this actually called a heteroduplex, right? Because you see in this scenario, the G come to with T, A come with C. They cannot match each other. And this mismatch will be recognized by the molecular cells, the cells system. The cell actually has this kind of power. Very often we learn this one, right? In the other courses, right? The cell get the repairing, the DNA mutation get the repairing, right? So there's many ways to repair. Today we mentioned about two, gap repairing, mismatch repairing. How it get repaired, right? Here, if we look at the mismatch repairing, then the GT can be repaired in two different ways, right? Either become AT or become GC. Similarly, the AC can be repaired in one of the two alternatives. A become T or G become C, right? So because of this, each mismatch have two possible outcomes. Then eventually you have 4 different combinations, right? In this scenario, if they both become G and C, right? Then you see the bottom and up become same allele, In this particular context, if they become same allele, we call it gene conversion. So this gene conversion is a technical term and used in this very specific context. When we're going to indicate the 2 different alleles become the same allele, okay? This is a very specific term used in these specific conditions, okay? What do we mean gene conversion? Gene conversion means at the beginning there are 2 different alleles. Then after the events, they become same allele. So this is called a gene conversion, okay? And here, you see, even though it is repaired, then you have the G and the C, you have the A and the T, it is still 2 different alleles, okay? Even though the allele may change their position, that's 2 different alleles are still there, right? The 2 different alleles are still there, so that's low gene conversion. This is similarly, that low gene conversion, even though they may locate at different DNA molecules, okay? And then in this scenario, you see both become AT, then become similar allele, then called a gene conversion, okay? And this idea actually very much relevant to the gene editing you may have learned in other courses, and that's why I'm thinking the genetics actually become a foundation for you to learn other courses, right? And some of you already approached to me and asked me, okay, about this kind of detailed mechanism, but even though I can mention it, but I may not able to explain in very much in detail. For example, here you see this in the CRISPR-Cas9 mediate repairing or homologous recombination, it will use the guide RNA, which is shown here like gRNA, the guide RNA, and to the target site, and then you make cleavage on the target DNA, so you create double-stranded not double stranded. I like to read as a double stranded. Actually, it's not double stranded. It's a double strand. Okay, there's no ED. Okay, DSB is a double strand break. Okay, you see, there's two scissors to make two cars. You generate the DSB, right? Double strand break. Then with this double strand break, you may have a random insertion and a random deletion. They come together called an INDER. INDER actually is a combination of the two terms, right? In means insertion. DEL come from deletion. So they call it INDER. Is the sign, I believe, is very much the biologist created the English words, INDER. Okay, so actually to represent this kind of gap can get repaired by randomly delete some nucleotides or randomly add in some nucleotides. Okay, they call it INDER. Okay, they call it actually lockout. Okay, then you can also get repaired by so-called homolog repairing. You provide a homolog region which can target on this side. So this region shown here, right, is homolog to this region, and this region is homolog to this region. And through homologous recombination, okay, and this can fill in the gap, then you call it lock in. And then you actually bring your gene of interest to the target DNA molecules. So that's how you can do the gene editing, right? So if this gene is your gene of interest, right, you put this gene into a DNA molecule, and this DNA molecule is very often is a plasmid, like the F plasmid, right? There's many different types of plasmid. One is the F plasmid. Okay, you put a gene in here, then this gene through this kind of homologous region, this gene can get into this gap, then get into target DNA molecules, then it will form a DNA molecule contain your gene of interest. Then you so-called gene editing. So DSB and CRISPR-Cas9 mediated gene editing is very popular and widely used in many research labs right now. Okay, many research right now. So if we go later, you go to the higher level courses, for example, in the 4,000 level in AOS, or even 5,000 level in AS, you will actually have the opportunity to do this one. Okay, do all this kind of gene editing through the CRISPR-Cas9. Okay, you have opportunity to do this in the lab. Then you will learn this much better, but I hope what we have learned here give you some basic idea. Then you learn, when you're going to learn the 4,000 level course, then it will be much easier for you. Okay, you look at the DSB, right? You know, right? What I mean, double strand break model. And at that time, you remember, Dr. Wu mentioned this one in his course. I will be very happy. Yeah, by remember DSB, you remember Dr. Wu. Okay, and also, right, there are many enzymes involved. Actually, the one of the, I think I know her by her voice, asked me, yeah, so, hey, what kind of protein are involved? What kind of enzyme are involved? You see, each stage, there's so many different proteins are involved. For example, this kind of, to create the double DSB, and to create the resections, and to stabilize the sticky ends. Once you create the sticky ends must be stabilized. And also, you must have those kind of motor proteins straight to move, right? Going to invade. And then this is the molecule you are going to, you provide, and then you wish to bring in, and you get into your DNA. So these molecules you provide, okay, by your own, and this enzyme you can purchase, okay, from a commercial company, and then you do all these things just in the small reaction tubes, and do all the stuff. Then you eventually get the gene edit, and then you, for example, you are interested to make, okay. And this actually happen, for example, very often happen naturally in our body cell, and it's one particular biological events, a very important Maybe I spent too much time to talk this kind of beyond the syllabus of this course. Any one of you are good at the immunology, and you learn how the antibody are produced. Anyone? Learn a little bit at least? Actually, you see the gene in our human genome encode for the antibody production. It's just one gene. But how many antibody we can create? Countless antibody. Even I don't say thousands different antibody, at least hundreds, right? We can generate new antibodies forever at one time, sooner we're alive, right? Sooner we're survival, sooner we take the foreign proteins somehow into our blood, we're generating a different antibody. How it can be? How it could be? Imagine. Because the gene is the same, but every time if you have different antigens, you're able to generate different antibodies. What's the fundamental mechanism behind this phenomena? It is through this one. Okay, through this one. So later, when you go high level immunology courses, or if you want to learn this one, you maybe recall this, okay, how DSB and how this, okay, non-homologous ending joining can create the different version of the genes, and the different version of the genes going to be transcribed and make different antibodies. The one gene can make so many different antibodies because of this. A piece of slides showing here. So yeah, I don't want to go anymore. Okay, and I cannot finish today's lecture. Okay, I go very quickly about the site-specific recombination. Very quickly, and this is an example. For example, this is the homologous recombination, right? You have two homologous regions. After crossover somehow, you get into, and you remember you change the orientation, right? Yeah. And this one, you still have a very homologous region, but for site-specific, it's just a very short region of this one. Then in this scenario, okay, for example, if you cut this, right? You have this big B here, and you go O, P prime, P-A-R-T, come down all the way, P, then you come to this O, come to this B prime. So this is how the site-specific recombination get into another organism's genome. For example, the phage, right? The phage. For example, the phage genome, this is the lambda phage, then this is the bacterium, right? They can integrate it into bacterial genome through this kind of very small region of the homonode. Okay, actually it's identical. They call it attachment site. ATT comes from attachment. P here comes from phage. Attachment B comes from bacterium. So attachment region at the bacterium, attachment region at the phage. They are actually very small, short region identical. They have cast at this position, they have cleavage on this position, they have cleavage on this position, they have cleavage on this position. Then once in the cut, we just rejoin. Okay, and this card we're going to join with this one, and this card we're joining with this one. So it's just cut and rejoin. Then they make this kind of recombinant molecules. Okay. Of course, they can integrate, it can also come out. Okay, but when it comes out, they need another enzyme called excisionase to help them come out. Excisionase, right? You know the term, and then you know what the function. Okay. And this is another additional information, just make you imagine better. You don't have, but it's okay. So the phage, lambda phage DNA get into the cell, they circularized, become at the beginning, actually it's a linear DNA, they become circularized, and this is circularized, right? This is ATT, the attachment site, right? The attachment site, they get integrated into the genome, they become like this. Later, they're going to synthesize their own genes and their own genomes, whatever they wish to synthesize, then generate progeny phage particles. Yeah. Okay, with this, I give you a short break. I move to the last part of today's lecture. We see about the transposon and the transposition. Okay, I give you a short break. Hello. Yes. That's not well. It's many of you are still confused. Yes, you need to read something.
Wait, so
Yeah.
just to check,
Yeah.
this part
Yeah.
is a bit of recombination also because
And,
like it's gene conversion, but it's also like a bit
it is, yes, but this is recombination for this molecule. It becomes recombinant, right? Because this one changed, right? So you had the ideal composition different from the original ones, right? Because your small b changed to big B, right? So this is, but this recombinant, as I mentioned, is not directly from the crossover. It's from the repairing. So, yes. Yeah. The answer is yes. It forms recombination, but this recombination is not from crossover. It's from gap repairing. Yeah.
like I don't really get how like it actually migrates.
Mechanism is just to the molecules move them, keep the string actually swirl. So this string keep going up and this string keep going down. So let's go into this string right keep going up, this string keep going down, and then get this aggregation can happen. So the cross actually move to the right side. Yeah.
Wait, then how does vertical cut?
Yeah, vertical cut, you see, right at the beginning, right, this crossover, right, it's a horizontal cut, right, it's easier, right? Yeah, the vertical cut is when this bottom one is going to rotate become this shape, and then the vertical cut like this. Yeah, vertical cut like this. So it must rotate. Once it rotates, it becomes this conformation, this kind of conformation. You look at the video, you look at the video, you will understand better. Okay, the video I shows to you. Yeah.
What causes the thing to differentiate between whether you rotate 100?
This is really a good question. Yeah, I think when they're going to know they're going to rotate or not going to rotate is really nobody knows. Yeah, nobody knows. Yeah, who is the controller? I don't think it's by random. Maybe have some so-called mechanism behind, and who is going to regulate, but just we don't know. Yeah.
Then for this part here,
Yeah.
are you really understand like how like
Is it better maybe you go to watch the video and to read the slides again, then you come back for a while, okay? Yeah, okay.
This one it says there will be no genetic variation because there's variation in the heterogeneity.
Yes, yeah.
But it means, but it's still not a recombinant.
It's not a I don't consider recombinant, just variation.
So recombinant is only if
After repair.
the alleles change.
Changed, yes,
But
right.
if it's a heteroinflex region, it's not recombinant.
Yes, it's not a recombinant.
And
Because,
then you will eventually be repaired.
yes, because you heterogeneous region alone, you cannot call it as recombinant because you don't know the outcome of the repair. Right? The outcome of repair can actually restore the wild type. It becomes the wild type, right? There's no anything change. But it also can come as become a recombinant. Yeah, so that's why at this stage we cannot call them recombinant because we don't know what will happen after repairing. Okay? Yeah. Maybe can give some chances to others. Okay. Yeah. It's just this why is 3' off? This maybe just the protein which binds to the 3'. The 3' is decays, right? There's only those 3' decays will bind by some protein. This protein actually can maybe drive the inhibition. Okay. Yeah. Sorry? Yes, not general, yes. So now, actually all. Yeah.
Yes, yes, right. Yes, right. And you only cut one string. Yes. There's all these string and this string, it's all cut these two strings. Yes. Yes, there's definitely some particular size for them to take a cut. Yes. Yes, right. So this part actually, this part actually not join anything. This part actually not cut, there's no join at all. Yes. All the part. Yes. Right. Okay. Give chance to this.
Oh, sorry. It was in one of the tutorials.
No need.
And I was like, okay.
This is a little bit complicated.
Okay, so ignore this. Yeah, I think that's it. Sorry, question. I did. Do you have a question? Oh, I was just wondering if I need notes. Because I was looking at one of the videos, and I was like,
This depends on how they make it. At the beginning, of course, they just scream, and later before this section, this one,
so I'm like, oh,
they
beautiful.
remove something.
I don't know.
The resection actually delete out.
Yeah.
The resection means it's going to delete out short. Yeah. The form is speaking.
But it's like worse because it doesn't have a tool, and I can't do like I can.
Yes.
I could, but I broke it. I broke it on day 2. It's crazy.
It's reverse this one.
It's okay.
Then they reverse this one, they reverse this one.
No, I feel like it's like nicer though because
Reverse.
like I'm lugging this thing around and I'm complaining that I have back issues. Like,
It's neither reverse. If we reverse this one, there's no reverse. Only this reverse.
I'm
Why
excited.
is start on cross
Honestly, if I didn't see I would need 3 things. I'd be like,
yeah,
laptop there, the camera in front of me.
So the cross start on the cut. Yeah, if both cross finger cut, this cross, both are cut, then it comes. So cut is not solved. Cut is just bringing them to join. And the cut but not solve any.
No, you count. So what you give them?
The further thing only get repaired at the later stage. Yeah, after this. No, the junction at this junction. Yeah, so this junction is and this junction is half, and this one, only this junction is half. This will come cross to E, and this cross is in only half. And this cross actually can be resolved by the K. And this is what I am showing you. This cross is half by the K. So look up in this.
Okay, let me continue. We move on to the transposition. And it's also very important because this transposition will create the genetic variation, create the distability. Okay, because as I mentioned, right, in the topic 4, we are actually trying to introduce the ideas, the knowledges, how the chromosome, the genome can be changed in our cell for good or for bad. Okay, for good and bad, right? In all the previous talk, we look at how it can maintain, the integrity, the stability can be maintained. But in today's talk, we look at the mechanisms, how the variations can be introduced, right? The recombinations and the transpositions. Okay, there's a three general pathways for transposition: simple transposition, replicative transposition, and retrotransposition. And this is caused by jumping gene. Actually, at the very beginning, the student also argued with me, Dr. Wu, you cannot pronounce the jumping very well. The word I pronounced always miss out the syllables or the super nice jumping gene. Okay. Then, if you look at this very interesting phenomena, right? It's because the jumping gene help us to lose the tail, right? If you wish, then you blame the jumping gene. If you appreciate, oh, it's good for us without tail. Hey, do you wish you have a tail? Do you wish we have a tail? You think that with lots of tails is good or bad? Naturally, I think we may have a tail is good, right? As humans, do you think with a tail is better? But anyway, it's just funny stuff. And then this kind of phenomena, right? There's even very interesting kind of the transposon is jumping gene. The name of the jumping gene is another name called the transposons. They actually have the power of viruses. They can come out, then to form the virus particles. That's how it looks like a virus particles, then jump from one cell to another cell and to create the gene variations between the different cells and even different living organisms. And this jumping gene is identified from, very recently identified from this kind of roundworm, okay, nematodes, okay. And this is a jumping gene. It's in this region. Let me draw you a pen. Actually, this entire part is a jumping gene. They can jump, okay, from one genome to another genome, from one cell to another cell, one living organism to another living organism. So it also cause the horizontal gene transfers and also cause the genetic variations. In one pathway is called a simple transposition, and that is very easy to understand. You just cut and paste, right? You cut out from this position and you get insert in another position. TE comes from the term transposable element. Transposable element is also the name of the transposon. Okay. Then very often for this element, they have the so-called a region called a DR. It's a DR. Then it's called a direct repeats, which are exact same sequence. You have this region, okay, and this region are exactly the same. They actually become a footprint of a jumping gene. And in between, actually it's a TE component. You also call an insertion element called IE, or it's called it's a simplest transposable element. Okay, in between you call it IR. IR called an inverted repeat. What mean the inverted repeat? If you look at the sequence where you look, right? So here is a CT, GA, CT, CTT. It's a 5' and a 3'. Okay, then inverted, what I mean, how to invert? If you look at here, right? This is 3' and this is 5', right? Then you have this top strand become here. Then come CT, GA, CT, CTT. So there's an invert, right? So here is on top. You read from left to the right, but here, right? You're at the bottom. You're from right to left. So that's how, what mean a reverted sequence, okay? And then in between, you get another enzyme. It's essential for them to jump around. So it's called transposis. Transposis. Okay. Then you have someone is a little bit more complicated jumping gene. It goes through the simple transposition in this scenario, right? Except for the essential gene, they carry some other genes. In this example, it's antibiotic resistant gene. It helps to spread the antibiotic resistant gene, right? So, but be mindful, only this IR, right, are involved in the transposition. So this box, the box region is considered a TE, okay? It wouldn't use this to IR, okay, this to IR. Okay, even though this to IR, but it's not function and not facilitate the jump from one place to another place, okay? It's only this to IR I highlight as this triangle, okay? Pay attention to, okay? So showing here, right? They show similarly. They show this kind of from here to here is considered a transposable element, considered a TE, and then you have this highlighted part as an identical repeat, right? It's called a DR. Direct repeat is identical sequence, just at two ends of the transposable element. Let's look at this video. I think you understand. I hope I have the video to show you. Yeah, you watch the video, you'll understand much better, okay, about this, how it works. Let me have the arrow, then.
The most direct mechanism for movement of transposable elements is called conservative or simple transposition. In this process, the transposable element is removed from its original site and transferred to a new target site. The transposable element is removed when the enzyme transposase recognizes characteristic inverted repeats at the ends of the element and cleaves the DNA at both ends, releasing the...
Remember each single word the video says here. It's very important for you. Yeah, again, this is a very important part. Okay, let's just go back. As said, right,
from its original site and transferred to a new target site. The transposable element is removed when the enzyme transposase recognizes characteristic inverted repeats at the ends of the element and
each single word. Okay.
Moved when the enzyme transposase recognizes characteristic inverted repeats at the ends of the element and cleaves the DNA at both ends, releasing the transposable element from its original site. The transposable element stays attached to the transposase enzyme, which cleaves another DNA segment at a particular target sequence. The cleavage at the target sequence produces staggered ends. The transposable element is inserted between the staggered ends. Then DNA gap repair synthesis fills in the base pairs in the staggered ends. The most direct mechanism, the transposable element winds up in its new site between two direct repeats of the target sequence.
So this is how the direct repeat are generated, right? It's when they insert in, right? They generate this kind of the staggered ends, right? Let me see. They generate these staggered ends, right? Insert in, then this gap was synthesized by this tape, by this template, and this strand was synthesized by use this template. So then that's why it become identical. Okay.
Then DNA gap repair synthesis fills in the base pairs in the staggered ends. Thus, the transposable element winds up in its new site between two direct repeats.
So that's why the DR, direct repeats, is become a finger mark or footprint, right? For example, later, if you are learning computational biology, then your professor asks you to identify, give you a DNA sequence with thousands base pair, then ask you to write a program to identify any transposable element in the genome. Then one of the very smart way is to write a programmer which can identify the DR which can identify the DR. So you have the identical sequence in somewhere, then in the between, it might be a TE, a transposable element, right? So this is actually how the bioinformatics is going to analyze the genome, analyze whether it contains the TE or not. That's actually why the genetics become a fundamental courses, right? So later you will learn the computation biology.
Of the target sequence.
Okay, that's all. Then if you go through the replication transposon, it's good. It's a copy and a paste. You copy the TE and insert it into another place. So it's a very smart way to amplify it itself, but it is a plagiarism, right? So don't do this when you're doing assignment. Okay, it's just copy from somewhere and put to in your assignment, right? So it's a plagiarism, right? And this one is a retro transposon. It's even more good way to amplify. You're going to synthesize the RNA and they use the RNA as a template to make a DNA, and they use DNA to put it into a new place. Because you have two ways in this step, right? One RE can make many copies of the RNA, and each RNA can make a double-strand DNA, and they put it. So this actually is a much more efficient way to amplify this retro-transposable element called RE element. Okay. In this example, this is one of the retro element called RE, is called allele. You see, this single transposon take about 11% of our genome. 11%. So far, we still get somehow intriguing or curious why this single transposon element is amplifying in our genome. What's their biological significance? Or they just somehow like bugs in our genome play around with us, play around with our humans. Right? What's their biological significance? Why, for what kind of purpose for our genome has this kind of 11%? Just like a jumping gene. We don't know their function at all, or at least not clearly knowing. Okay. And then when we study this jumping gene, may actually help us to dealing with the HIV infections, because you see these jumping genes requires the enzyme so-called integrase, right? You replicate the DNA, they integrate another position, involve an enzyme called the integrase. So if you study the integrase function and they identify any inhibitors can inhibit the integrase functions, then you can also use this kind of inhibitor to treat the HIV infections, because HIV infection requires this enzyme for their own replication. So all lineages actually is linked. Okay, you link to each other. Then come to the second important part of this topic. Okay, we have two focus for today's lecture. One is DSB model. The second is this part. Okay, transposons become important tools in biology. It's called a transposon tagging. And it become important, be honest, it become less and less lab are use this methodology. In the older time, actually, I use this methodology. Okay, but right now we have more advanced biotechnology tools to edit our genome. Okay, but in the older time, it's still using, some labs are still using, but be honest, it becomes less and less. Okay, but then the biological phenomena is interesting. For example, how we can identify a gene involve this kind of eye color involve this eye color, we can introduce a transposon into the embryo of flies, and to look at which flies get lost the eye color. Okay? If we lost the color, we call the loss of the function mutations, right? Loss of function or mutations. And this transposon we call the transposon mutagenesis. We introduce the transposon into the embryo and hope the transposon become a mutagenesis to mutate the gene. So, of course, it may create random mutations, but my interest, for example, in this particular example, I just interested at the eye color. It may cause other mutations. I just throw it away, trash it, right? For example, other flies may lose their wing, other flies may lose their maybe necks, maybe lose other part or the hair. It's okay, but it's not my interest. My interest is just like the eye color. Then we can, once you find this phenomena is in my interest, oh, this fly will become my baby. Oh, I finally find a fly with the white eye. Then you treat this white eye very preciously, take out the cell, and get the isolated genome out. Then you use the transposon, right? Because you know that which transposon you put in, and then use this transposon as a tag. That's what I mean, transposon tagging, right? And then use the transposon as a tag to identify where the transposon it is. For example, if you identify the transposon in this region, and then you believe maybe the region around this transposon maybe is the gene for eye color. Because you jump in and they disrupt this gene function, and then you believe this region is related to eye color, right? That's why it's called loss of the function mutation. Because they jump in and they disrupt this gene function, and that's why they lost eye color. And of course, you can use many later laboratory skills to demonstrate and prove it is true, right? You can use PCR, you can sequencing, you can use cloning, can be used to verify the gene function and to verify what you have identified. But the idea it is you track your transposon and find where they are, then you make an assumption the gene around the transposon maybe is the gene associated with the eye color. Then you isolate it out, you can study and to verify whether your hypothesis is true, right? And this is what, for example, the student learn this part. I want to show you what is a good way to learn, right? After you learn, you can internalize what you have learned. You put a sequence of how the transposon, like the transposon name is copia, is the name of the transposon, how it works to identify the eye color genes, right? So the step 1, 2, 3, 4, right? Okay, towards end of the course, not the course lah, end of my lecture session, I want to show you some of the philosophy I used in my teaching. For example, anyone can take the riddle. What's the first one? I forgot to bring some kind of the candies. Okay. Anyone? Can you break the riddle? Windows. Put hands to her. Around the floor. Yes, window. I wish to use this one as an analogy. What you have learned here is just a window. It's a small, tiny part of the entire knowledge of the genetics. Okay, I hope you can open the window, then you get attracted by the beautiful of the sceneries out of the window, right? Then you jump out, you walk out of the room, and to explore on your own. But what you wish to emphasize here, what this? What's the, anyone? Again?
To learn for life.
To learn for life, yes. Give it, man. Put our hands together. Yes. To learn for life. Don't to learn for exam. To learn for life, you're enjoying the learning. If you just learn for exam, you're you're stressed. I think you'll be stressed. Okay. So there's another homology, right? Homolog, 2 and 2. 2 and 2. Okay. So this is more fun. What means class? Come late and start sleeping. And what is life? School? 7 creepy hours of our life. Wow. Yeah, this is another one, right? To look at the biological phenomena, I just want to show you what you have observed. For example, as shown, right? This kind of jumping gene, the mu get jumping in, so the color get lost. Then when it can jump in, it can jump off. Once it jumps off, it gets restored, and then in the cell, they jump off, they can produce the eye color. Oh, not eye color, sorry. This kind of pigments in the cone, right? Or in the other whatever. Right? So this shows why you can observe kind of mosaic. Because you observe this kind of mosaic, because this cell, for example, this cell can generate pigments of a color, because you can imagine, because this guy jump off, the gene function gets restored. And this one is happened to all of us. When we are young, this jumping gene actually was silenced, was trapped in this kind of highly condensed chromatin, heterochromatins. You see, the chromatin, as I mentioned, need to organize in the 3D structures, highly compacted, well organized. All this kind of organization requires what? Energy, right? If you link the philosophy or knowledge of the physics of entropy, right? We need a lot of energy to put everything in order. If we lost the energy, when we become older and older or something goes wrong, we actually are unable to maintain this kind of highly compacted heterochromatin structure. Then those kind of monsters, I'm not in tighter control, then I'm going to jump. Once it's going to jump, when aging, they jump to the different places, then cause different troubles. You get a new insertion at many wrong places. That's why when older, we get more and more weaker, get more and more disease, and even more cancer. Right? Cancer very often much more occurrence. Right? The rate is much higher than the others. It's one of the reasons it is. Who knows? In one of the jumping positions, the cell lost the control, the cell cycle control, then going to amplify. Then lost the cell control, then become. That's why when older become a lot of disease. So I'm imagining a lot of jumping genes in my body that are jumping around. Yeah. And this is one, right? They bring the gene to many different organisms. And the summary, all the 4 topics: chromosome, and chromosome. Okay? How do we maintain chromosome, and how chromosome may change over time? And if we look at the history, the people working on the chromosome, how many Nobel Award given to? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, and going on. Or actually, this highlighted are all Nobel Awards. Okay, so there's many places for you to fill in and to sit in to get a Nobel Award. Okay, if you're interested, look at all the study here. And last but not be the least, I want to use this example, right? How the genetics may be relevant to your physics, psychology, and I know some of you actually study psychology as a major or second major, or you are in a psychology major, you learn life science as a second major. You see here, now what happens is, use the mouse model we found. If we allow the mother mouse and the baby mouse stay together, they leaking and grooming, then it will affect the epigenetic markers, which here shows the methylation on the DNA. And then it will shut down the gene expression here, and eventually will help us to manage the stress. So this kind of gene expression going down, and then actually it helps us to control the stress. And okay,
Oh, sorry. This is a lure. Okay. Lure leaking and grooming. Actually, then the stress were going up. Okay, and the high anxiety. So this gene is pressed down. If this gene gets shut down, then this gene gets low expressed, then you get high anxiety. If lower, but if the higher, the baby mouse and mother mouse they still together, then this can switch off. When this methylation group gets removed, right? Then you get lower anxiety, right? So use the mouse model as an example to learn you as a mother's behavior when you have your baby, right? In the morning, you have to go to your office to work, in the morning to buy the milk for your baby. But you look at it, remember this image, right? Before you leave, you better leak in your baby, groom your baby, right? To remove the stress. Human leaking is just kissing, right? So leaking and hug, grooming your babies, right? So to make sure that genetic modification is a favorite to lure their anxiety, right? Your baby wouldn't crying, right? So I wish you all enjoy the learning and develop something even more I desired and hope for. And this is another piece I get a little more time to share, but I really like this kind of thing. Maybe I just, hopefully, I get. Can you bear me with a few minutes more?
That's right.
No, this one. Where's my...
The false belief.
This is actually a guy from in the area of the architecture, okay.
That children learn best in quiet places. Completely wrong. School should be where the action is. School should be right in the center of the city. Think of the old, the Raffles Institution,
SJI,
ACS. When children
move on
in their
quickly and move on to the
years,
part I
live
wish
close
to
to where the social actions are, the cultural activities. The issue of diversity is even more important in a small, unified state like Singapore. Because
diversity.
The biggest problem in Singapore is human resource development. Guan Yu has always lamented the fact that we don't have enough talent because our population is small. Therefore, we need a lot of foreign talent. I agree with him. We need foreign talent to some extent. But more importantly, we need to develop local talent. Epigenetics, which is the new science which has been around for the last 15 years, right, is a very important contribution to genetic thinking. Epigenetics is the part of the human genome that switches on or off various genomic capacities. Switching on and off in epigenetics is conditioned by the environment. Therefore, there is an interconnection between how you plan your physical environment and how you run the society, right? That means the hardware and the software that will switch on or switch off various kinds of capacities. My argument is that we are not switching on enough of our creative capacities. That's a point.
So you see this kind of architecture, this kind of politician, they are able to use the genetic knowledge to verify or to convince people the way of thinking, the way how you should build up our society, how you should build up buildings to affect to bring the beneficial to our societies, right? Okay, this is the one last piece of slides. Can you identify in this conversation what are genetics, what are epigenetics and genetics, right? In this example, the Uguisis, let me show you. I hope.
That's right.
You look at this one.
Friend, the panda will never fulfill his destiny, nor you yours, until you let go of the illusion of control.
Illusion?
Yes. Look at this tree, Sifu. I cannot make it blossom when it suits me, nor make it bear fruit before its time.
So this actually, they talk about genetics, right? We cannot eat blossom, it suits me, or make it bear fruits before the time. This means they're controlled by genetics. Then
there are things we can't control. I can control when the fruit will fall. And I can control where to plant the seed.
That is no illusion, Master.
Ah, yes.
There are something we can control. We can control the environment where sow the seeds, right? This is very obvious. If you have seeds, you put on the table, they will germinate, grow a plant. But the same seeds, if you grow and sow in a suitable environment, it will germinate, right? Same as what they argue here.
But no matter what you do, that seed will grow to be a peach tree. You may wish for an apple or an orange, but you will get a peach.
But
So this comes back to genetics. You cannot expect a peach tree to give you orange or apple, right? So this actually genetics area in our daily life.
Maybe it can, if you are willing to guide it, to nurture it, to believe in it.
But how? How? I need your help, Master.
No, you just need to believe.
I believe you.
Promise me, Shifu.
I have my great appreciation to all of you. Thank you for coming to support my teaching. Thank you. And we all the best in the CA and in the later part of your journey. Okay? With NS.